I am really happy to have the opportunity to speak in front of this group today. And I'm going to be talking about RNA genomics technologies and how they can facilitate RNA research. So first, just a really brief background on Eclipse Bio. So we were founded in 2017 with the mission to develop novel RNA genomics technologies that facilitate and accelerate RNA research. But perhaps more importantly, our mission is really around making those technologies accessible. So we develop kits and we offer services, and also we're developing data analysis tools. So let's dive into the RNA world here. So what I have depicted here is just basically an embellished version of the classic central dogma: DNA transcribed into RNA and translated into protein. But as you can see from the diagram, the RNA world is actually just much more complex than just a simple linear molecule. And as you can imagine, the traditional RNA-Seq is very unidimensional relative to really what's going on in the RNA world. So I want to talk a little bit about levels of regulation in RNA, and then we're going to dive into some specific genomics technologies that look at these different levels of regulation. So I think you're probably all familiar with differential splicing that yields different RNA isoforms. But there are many other levels of regulation that can currently be addressed with NGS-based methodologies. So I'll be talking about those today. So first off, RBPs, these are RNA binding proteins, and these are shown here in red. And RBPs can regulate many aspects of the RNA life cycle. I think there's around between 1,000 to 2,000 different RBPs that have been identified. So there's just a myriad aspects to RBP regulation of RNA at the level of stability, at the level of translation, and structural motifs as well. Turning over to modifications here, there are actually over 150 different RNA modifications that are known. And I think we're just sort of scratching the surface of really understanding the implications of the epitranscriptome. So this is really a burgeoning field that I think will be growing quite rapidly as tools come online to really look at these different modifications at a transcriptome-wide level. So I'm going to be talking about one of those methodologies to look at M6A today. And down here in the right-hand corner, UTRs. So these are the untranslated regions at the ends of the RNA, so the 5' UTR and the 3' UTR. And I think, as you're probably aware, the transcription start site can be a point of regulation. So transcripts can modulate which transcription start site they're using, and the same with the polyadenylation site. And so I'm going to be talking about a technology that looks at the mapping the TSS and the PAS with single nucleotide resolution. So over here on the top left, RNA structure. So as you're probably aware, RNA is not just a simple linear molecule. It forms complex secondary and tertiary structures in vivo, and these regulate many aspects of the RNA life cycle. So obviously, accessibility to binding with RBPs and microRNAs, and also it's a point of regulation basically for modulation by therapeutics. So small molecule therapeutics, binding various structural motifs. This is a burgeoning area of research. So I'm going to be talking about an NGS-based methodology to look at RNA structure. MicroRNAs, you're probably familiar with microRNAs being a major point of regulation, post-transcriptional regulation of RNAs. And I'll be talking about a technology that really precisely maps the microRNA-mRNA interactome. And finally, the translational control, the rate at which ribosomes load, the rate at which they translate RNAs into protein, again, is a major level of regulation. So we'll be going over some of these technologies today. Most of these are technologies that are already on the market or in the scientific literature. A couple of these are technologies that we have in development, but I'd like to tell you a bit about them just to give you a really more comprehensive view of what's going on in the field of RNA genomics. Okay, so before we dive into the individual technologies, I want to talk a little bit about RNA genomics and how it's used in both academia and industry. And so we work with a number of both startup biotech companies and big pharma, and also academic researchers. And so I think we have a pretty good picture about how these technologies are used in the field. And I wanted to give you just a little bit of overview around that before we dive into the individual technologies. So I don't think that the realm of basic biology needs any explanation here. RNA is clearly a major point of regulation in basic biology, development, disease, etc. And I think these technologies are really providing new tools to look at very subtle but yet important aspects of RNA. Over on the side of RNA therapeutics, this is a really rapidly growing field. There are now over 100 different RNA therapeutics currently in clinical trials, and just a growing number of startup companies that are tackling RNA therapeutics from different angles. And, just sort of broadly, RNA therapeutics strategies kind of fall into two main buckets, I would say. The first being mRNA therapeutics. So these are, that is when the goal is to express an mRNA in a cell or in an organism to replace a particular function that may be deficient or mutant. On the flip side of that, there's targeting RNAs to knock out a particular function, and the therapeutic modalities that are used in that realm are typically antisense oligonucleotides, or ASOs. siRNAs are also a main sort of therapeutic modality that's in clinical trials now. And also, I would say a growing area is small molecule therapeutics. So, traditionally, small molecules have been used to target proteins, but there's a really growing sense that targeting RNA allows the druggable space to really be increased. In other words, there's many more targets if you're also able to target both RNA and protein. And finally, I don't think that vaccines needs any background here. I think we're all well aware of the power of RNA vaccines and how quickly they can be deployed. And I think we're just really scratching the surface of what we're going to see around the corner with RNA vaccines, tackling indications sort of above and beyond infectious disease. And over here on the left, RNA biomarkers. So in the realm of diagnostics, looking at the gene expression levels is a modality that's been used in diagnostics that are on the market. But as new tools are developed around RNA genomics and more subtle aspects of RNA regulation are discovered, I think that there's going to be a growing sort of area of different types of RNA biomarkers that can be used to develop both diagnostics and prognostics. So now for this portion of the talk, we're going to dive into individual technologies to give you a sense of, both how they're done and how they're employed and what sort of data that you can get out of these types of technologies. So we're going to start off with e-CLIP, which is used to map RBP-RNA interactions. And this is actually the technology on which our company was founded. So e-CLIP is the origin of ECLIPS. And let's see, moving on to tell you a little bit about it. So this e-CLIP technology was actually developed by the co-founders of ECLIPS. So it was done in Jean Yo's lab at UCSD, Eric Van Ostrin, and Alex Shishkin, who are all co-founders of ECLIPS. And it's a methodology that's used to map the precise binding sites of RBPs on RNAs transcriptome-wide. And so it was basically, built upon a foundation of related CLIP type technologies that are all in the literature. And very specific improvements were added to, really refine that data and make it more reproducible and really more deeper, more complex data that really profiles the transcriptome more effectively. So just taking you through what an e-CLIP assay is and how it's done. It's based on a UV cross-linking step. So the UV is done while during the cell culture step, basically. So essentially that's trapping the RNA, the RBP on the RNA with that covalent cross-link. And following that, the RNA is fragmented. So now you have just that little piece covalently bound to the RBP. And at this step, there's a number of steps that really kind of increase the stringency of this assay and also add to the sort of the effectiveness of the data analysis. And that is that, basically the antibodies used to IP the protein, but also there's a input, which is not IP'd, that gives you basically the background. And subsequently that is subtracted out at the bioinformatics stage, but it really gives you, I think, an enhanced look at where these RNAs are minus background, basically. So there's a number of, sort of high stringency steps here in terms of, both the IP conditions, and then as it goes through a gel and a Western, which basically means that only the RNAs that are cross-linked to the protein are transferred to the nitrocellulose. The regions of the gel are cut out of the nitrocellulose, and then library preps are made. And this is done for both the IP and the input, yielding 2 libraries that are sequenced and analyzed in this manner. So you can see that the having that input control allows you to bioinformatically exclude these false positive reads. So you can see here where the input looks just like the IP in terms of the number of reads and the signal here. That's obviously a false positive read. Where over here you can see just this really big peak here for the IP, and with the absence of a peak in the input, and this would be called as a true peak. So at ECLIPS, we've extended this to, many different proteins. And I just wanted to show you an example of one that is particularly interesting. So ADAR1 is RNA editing enzyme, and it binds ALU elements and double-stranded RNA, transcriptome-wide. And what you can see here is that we've eclipped 2 different isoforms of ADAR1 and found that they actually bind different elements. And so here the P110 is binding ALU elements, as you can see here, And the P150 version of this enzyme is actually binding line 1. So I think there's, a really interesting aspects of the ECLIP data and nuances that you can see with this level of resolution. So in fact, you can see even in the pattern of the reads binding here, you can see the structure of the RNA sort of recapitulated in that mirror image here. So the founders of our company published a paper in Nature profiling over 150 of these RBPs in 2 different cell lines, and then uploaded that data to the ENCODE database. So if you're curious about what eCLIP data looks like, or if some of the proteins that you're interested in studying are profiled, you can check out the ENCODE database and download some data. This is just kind of a overview of some of these different RBPs and how they tend to bind different regions of transcripts. So some of these are intron binders, some of them are 5' UTR binders, et cetera. So eCLIP is a methodology that it was developed initially on cell cultures, but can actually be applied to tissues as well as postmortem tissues. So we've done a lot of work with many different cell types. So I'm going to move on now to the epitranscriptome and a variation on the theme of eCLIP that profiles M6A. So M6A is one of over 150 different RNA modifications. So as I mentioned earlier, the epitranscriptome is actually very complex. And as I also alluded to earlier, I think we're just starting to scratch the surface of it. This is just a little peek at the publications around just M6A. With over 1,500 publications just last year, but you can see it's growing exponentially. And I think that really is a reflection of the tools that are available and our ability to look at what M6A is up to. And this diagram just gives you a sense of the areas of biology where M6A has been implicated. It's basically implicated in development, disease, and all sorts of aspects of RNA regulation. I also want to mention that, M6A is known to be regulated by writers, erasers, and readers. And these are all RNA binding proteins, or RBPs. And we can actually eCLIP these writers, readers, and erasers. And so, if you're interested in studying M6A, you can tackle it from multiple angles, basically, looking at the RBPs that recognize the modification, but also mapping the modification itself. So this is a look at the M6A protocol, and it's basically a variation on the theme of eCLIP, except that the RNA is first isolated from the cell. So this is, RNA that's been purified, and it is IP'd with an antibody that really specifically recognizes that M6A modification. So that antibody is recognizing the RNA itself, and the cross-link step takes place after that IP. And so basically we're cross-linking the antibody to the modification. And then it's taken through, these sort of stringent purification steps of eCLIP, and a library is prepped. So I think if you really want to understand at sort of a schematic level what this technology is doing with respect to the NGS reads and this cross-link and so forth, I think this picture really sums it up nicely. So again, the antibody is directly cross-linked to the RNA. The M6A modification is known in the literature. There's many papers on the Drach motif So that's this motif shown here. And so the modified adenosine tends to be found in the context of the Drache motif. And so you can see here that that cross-link of the antibody to the RNA, when the RT comes along, it tends to fall off. And, you know, we've actually kind of developed some more nuanced bioinformatics pipelines around mapping that cross-link site. But this sort of, you know, sums up essentially what it's doing is that it's mapping that cliff that occurs when the RT falls off the transcript. So looking over here at what the data looks like, you can see that a classic miR-eCLIP, which is just sort of the IP without the cross-link, gives these sort of more diffuse peaks with no resolution of where those sites are within the peaks. Whereas the M6A eCLIP, this might be a little hard to see, but you see these little cliffs here, and those are called by the algorithm as single nucleotide resolution M6A calls. And so that's this track down here. And shown here is the Drache motif, which is actually relatively common. But transcriptome-wide, you know, greater than 50% of these mapped cross-link sites tend to be in the adenosine of the Drache motif. So I wanted to give you an example of how these technologies are used in basic biology. And actually, this is sort of a preclinical therapeutics development here. This paper is an example of using a number of these RNA genomics technologies. So what they did here was a CRISPR screen to investigate the function of RNA binding proteins, or RBPs, in MYC-driven breast cancer. So essentially the question they're asking is, you know, when we knock down individual RBPs, which ones have the effect on the proliferative capacity of these cells? And they identified YTH-DF2, which happens to be an M6A reader protein. So they identified this as being very important for MYC-driven breast cancer. And in this paper, they you know, go on to you know, substantiate this with animal data, et cetera. And there's actually now a startup biotech that is you know, furthering this discovery as well. But as you can imagine, just based on the fact that this is a RNA binding protein and an M6A reader, these technologies really come into play to really investigate the nature of a result like this that comes out of a CRISPR screen. So this is so RBP-eclip was used directed against the YTH-DF2 to define the binding sites transcriptome-wide. And M6A eCLIP was used to investigate the M6A modifications in this publication. Also, this is also a kit that Eclipse has on the market. This is not a RNA genomics technology. It's actually a library prep technology that facilitates the preparation of libraries from CRISPR screens. I'm not going to be talking about this technology in detail today. If you are doing CRISPR screens and interested in you know, a simpler methodology for library prep, do check that out on our website. Okay, so continuing our survey of RNA genomics technologies, I'm going to move on to yet another variation on the theme of eCLIP, and this is to look at microRNAs and the interactome of microRNAs with mRNAs transcriptome-wide. So miR-eCLIP is a methodology that generates these RNA binding maps or interactome maps in a manner that's relatively unique and very precise, actually, as you'll see when we take a look at the data. And at its core, it's a eCLIP. And again, eCLIPs are always mediated by an antibody-based methodology. So there is a IP directed against the AGO2 protein. So this is your RISC complex here. That is where the microRNA-mRNA duplexes are processed. And so that complex there is IP'd. And then there's a ligation step in vitro. So ligase is added, and this is basically a proximity-based ligation that ligates the microRNA literally to its mRNA target. And I should mention, I'm kind of skipping over a few steps here, but the RNA is fragmented. So now you've got these little pieces of RNA where the microRNA is bound, and these two RNAs are ligated together. Now, ultimately, that goes through a library prep, and what it generates is what we call a chimeric read. So a microRNA fused to its mRNA target. And this library structure is actually very useful because, as you can imagine, I think you can see that now you can look at many different microRNAs and where they're bound transcriptome-wide. So, one of the really enticing aspects of this methodology and the data, I actually find this data really, really pretty to look at, is that it's very high resolution. So what I'm showing here is the 3' end of the E2F3 gene and a sort of classic AGO2 E-CLIP. And as you can see, AGO2's all over the 3' end of this particular transcript, probably reflecting quite a lot of microRNA action there. And this is the total miR-eCLIP. So these are the chimeric reads. And as you can see already, you can see that the resolution is really quite enticing, even though these are all of the microRNAs all together. But what's so great about that chimeric read is that you can deconvolute individual microRNAs and their binding sites. So that's what's shown here, is that you can look basically underneath these peaks at individual microRNAs and their binding sites. So this is a really interesting and precise way of looking at microRNA-mRNA interactions. I should also mention that, because siRNAs, and again, this is a very commonly used therapeutic modality, siRNAs are also processed through the RISC complex. We can also use the same methodology to map siRNA-mRNA interactions. And as you can imagine, typically with an siRNA, you're really targeting that toward a specific transcript with the intent of knocking it down. But this type of methodology, where you're able to look at the whole transcriptome, can effectively map all of those off-target effects of these siRNAs. And so we think that's going to be a really interesting application of this methodology. So another one of the really appealing aspects of this type of data is that we found that it is quantitatively related to the extent to which the mRNA is downregulated. So let me walk you through that. In this experiment, miR-124 was transfected into cells, and then there were two assays run. One was the miR-eCLIP, the assay I just showed you, to look at the transcriptome-wide binding sites of this miR-124. And then an RNA-Seq was also done to look at gene expression changes induced by this transfection of this microRNA. So over here, now this is showing the gene expression level. And down here, what we're looking at is different numbers of chimeric reads mapping to particular transcripts. But first, let me walk you through this. So transcriptome-wide, most of the genes are not changing with respect to gene expression following this transfection. if you analyze this data by, if you're actually look at target scan and the predicted, so these are bioinformatically predicted target sites of miR-124, you can see that many of those target sites are actually downregulated, many of those target transcripts actually. However, the data, the targets identified by miR-eCLIP actually give you, I think, a more nuanced and precise and quantitative view of the extent to which these targets are downregulated. So the targets that have greater than 3 chimeric reads are somewhat similar to what you might predict bioinformatically. If you filter for greater than 10 chimeric reads, you're seeing that these transcripts are tend to be downregulated more. And again, kind of underscoring the quantitative nature of these chimeric reads, the transcripts with greater than 25 chimeric reads mapping to that particular target, again, are more extensively downregulated. So kind of in addition to sort of the precision of the mapping itself, I think this sort of quantitative nature of it, again, I think, gives greater insight into what's going on in the microRNA-mRNA interactome. So we've developed a couple of different variations on the theme of this miR-eCLIP technology that really allows you to look really deeply at either a particular microRNA of interest or a particular mRNA transcript. So, and maybe I should back up for a moment and say that, as many of you are probably aware, transcriptome-wide experiments of any nature really tend to, profile the most abundant transcripts, the best. And some of the more rare transcripts may not have the depth of data that you may be looking for. So in the case of the microRNA interactome, we've developed 2 different variations, main variations on the theme. We can enrich for either a microRNA of interest or a gene of interest. And by extension, also an siRNA of interest, which is very similar to enriching to a microRNA of interest. These schematics are a little bit complicated, but I think if I just mention, looking at the structure of the library itself, essentially these enrichment methodologies are done at this level where we're using a probe-based enrichment strategy to enrich for either a particular microRNA of interest based on this chimeric read or a particular mRNA of interest. And again, this yields really deep data. And so the interesting part about this is that, we can multiplex different microRNAs, or we can look at, relatively similar microRNAs by enriching for an entire family of microRNAs. And essentially what this does is it really allows a level of depth in the library that's generated to very deeply profile a particular microRNA. And what that means is that you identify more mRNA targets for that microRNA of interest. And this is an example of the flip side of that. So enriching for a particular gene of interest. And in this case, we've enriched for the APP gene. And what we're showing here is just the 3' UTR of the APP gene. And shown here are the various elements of the data that have been pulled out. So again, the generic AGO2-eCLIP, which again is more diffusely over this 3' UTR. And then down here are these individual microRNAs. And again, since we've enriched for this gene, we're getting this really, really deep profiling of the microRNA population that is regulating this particular gene. And what we've done here is color code the related family members. And so, as you can see, some of these related family members they may differ by only a nucleotide or two from each other. They share the same seed matching site. So down here, we've color coded the seed matching site. And, what we've found over the course of many experiments is that, these microRNA peaks tend to be basically right over the seed matching site for that particular microRNA. And you can see that, multiple family members that share the same seed matching site are essentially sharing the duty, if you will, of binding that particular seed site and regulating this microRNA. So when you do this deep profiling of a particular transcript, you get this very nuanced view of sort of the microRNA world that is regulating this transcript. And I think, when you look at data like this, and especially these related family members, you can start to understand some of these classic results where, knockdown of an individual microRNA may not have a phenotypic effect. And you can kind of understand why here, that if multiple family members and multiple microRNAs are really sort of sharing the regulation of a particular transcript, you can see why just knocking out one of the family members may not have that phenotypic effect that, you might anticipate. Okay, so I hope I'm not going too quickly through these technologies, but I do have a few more to cover. And I want to talk about End-Seq, and this is a methodology to map the ends of the RNA and to really look at the UTR landscape transcriptome-wide. So again, what we're looking at here is the transcription start site for the 5' End-Seq and the polyadenylation site for the 3' End-Seq. So essentially what we're mapping here is that junction between the 3' UTR and the beginning of the polyase site. So let's see. So let me just dive into what the data looks like. Let's start over here. This is 3' End-Seq. So I should mention these are two separate protocols, two separate kits, if you will. And if you look over here at the 3' end, these are the annotated transcripts for this gene. You can see there's 4 different annotated polyadenylation sites for this transcript. And what we're seeing in this particular cell line is 3 of those are being used. And so this End-Seq data basically piles up the reads right at that junction of the polyadenylation site. And the bioinformatics pipeline that analyzes that data gives a single nucleotide resolution end call. And that's what's shown by these black tick marks here. And I think it's also interesting, you can see the relative usage between these different 3' end sites by the height of the peak there. And shown up here is traditional RNA-Seq. So you can see the RNA-Seq reads are really, they're going to be spread out all over the gene. Whereas the End-Seq data, you're really concentrating those reads right at the 3' end. And I'll show you some more data about how those reads are distributed. But really what that does is it, raises the level of effectiveness of the data in terms of calling that 3' end. Over here on the 5' end, again, we're really concentrating those reads right at that transcription start site. And this is an example of a little bit more subtle data here. The bioinformatics algorithm, again, is calling those peaks here and with giving those single nucleotide resolution calls here. And you can see, we see both of these annotated transcript isoforms here for the 5' End-Seq. So shown here on the left is a metagene plot, which many of you, I'm sure, are probably familiar with. And if you sort of look transcriptome-wide, this is a, sort of a kind of a view of a condensed view of genes transcriptome-wide. So with a 5' end and 3' end shown here, Typical RNA-Seq is going to be spread out all over the gene very evenly. And shown here is the enrichment of the 3' end in the 3' End-Seq data. And over here is the enrichment of the 5' end in the 5' End-Seq data. So you can see you really get a lot of sort of bang for the buck, if you will, in terms of the amount of data that's really piled up at the either the 3' end or the 5' end. And that's what's shown here with these distribution plots as well. So there's multiple uses for End-Seq, and as I mentioned earlier, we work with a number of RNA therapeutics companies, and I think the UTRs tend to be regions where there's structure and also regulation, both at the level of microRNAs, at the level of ribosome loading. But also this is a region, I would say, of high interest to entities that are interested in targeting particular RNAs with small molecules. And so that is actually one of the uses of it, is really precise UTR mapping in cell lines of interest or RNA populations of interest in the context of drug targeting. And also this can be used with respect to biomarker identification, so differential end calls with respect to disease states. And, I think also, let's see, so, different isoforms, of course, I think with respect to different transcription start sites or TSS and polyadenylation sites. So I'm not going to take you through this workflow in detail. I'm just going to mention the basic element of the protocol. All of this is on our website if you really want to dive into it in more detail. But basically for 5' End-Seq, there's sort of an indirect selection for the cap structure. And so we're employing a 5' exonuclease that is only able to digest uncapped molecules. And so, post-fragmentation, the short RNAs that have the cap are the ones that are selected for in this protocol. And those are the ones that are enriched in the library prep. For the 3' End-Seq, there's also an exonuclease or an endonuclease. RNase-H is used in along with an oligo-DT primer to digest that poly-A tail, and adapters are ligated on, yielding that precise 3' end enriched in the 3' End-Seq library. So I think I've already pretty much gone over sort of the uses of the 5' End-Seq and 3' End-Seq. These protocols are actually relatively simple molecular biology protocols with a library prep. And so these are both available in relatively easy to use kits. So I want to talk a little bit about our technology pipeline at Eclipse and talk a little bit about some of the technologies that are right around the corner. And I think this will help give you a picture of, where RNA genomics is headed and kind of a more nuanced view of, what RNA genomics can do. So I mentioned translational control and looking at the translatome. And I think this is going to be a really, really burgeoning area of research. The loading and I would say the extent to which ribosomes load onto given RNA transcripts not only affects like translational control in vivo, but as you think about the realm of RNA-mRNA therapeutics or RNA vaccines, where you're introducing a synthetic RNA and you want to be able to modulate how well it's translated. You want translation to be efficient, so you want to make sure that you're able to modulate the structure and the modifications on that synthetic RNA such that ribosomes load efficiently, but not too many ribosomes so that you get ribosome collisions and stalling, et cetera. So being able to look at ribosome loading in a very nuanced way, I think is going to be important in the realm of therapeutics. And clearly it's important in basic biology. It's a really well-known level of post-transcriptional regulation. So RiboE-CLIP is again another variation on the theme of eCLIP, and I'll get to the sort of the schematic of it in a moment. But it basically is a methodology that is much simpler than traditional means of profiling ribosomes. So standard ways of sort of classical ways of looking at the ribosome load on particular transcripts is either polysome profiling or ribosome profiling, which is also known as RiboSeq. Both of these methodologies are kind of based on sucrose gradient sedimentation, requiring ultracentrifugation, fractionation, fraction collection, and sort of library prep from those fractions, et cetera. So RiboE-CLIP, and we've actually developed a simpler, easier version of a RiboE-CLIP, is a way to kind of skirt around some of these technical challenges and get this data around ribosome load. So again, I'm not going to go through the protocol in detail. You can check out our website, but basically, again, it's a standard eCLIP protocol whereby a antibody directed against a ribosomal protein is used to pull down and map those RNA fragments that are associated with the ribosomes. Now, in parallel, we're also doing an RNA-Seq, and ultimately the comparison of the level of transcript versus the level of transcripts that are associated with ribosomes gives you that translational efficiency. So this just shows you the structure of the library. And what this does basically is it gives you a sense of where the ribosomes are and the extent to which ribosomes are binding particular transcripts. So you can see here a traditional RNA-Seq. You're still getting reads in the 3' UTR, whereas when the ribosome drops off, you can see the diminution of the signal there. And really, I think one of the main uses of this type of technology is really to look at translation inhibitors in the context of either, sort of traditional translation inhibitors that might be used in sort of, oncology and therapeutics, cancer therapeutics, but also, really specific RNA therapeutics directed at particular transcripts and affecting their translation. Shown here is a classic, translation inhibitor, taurin, which is known to affect particular translation of particular genes without affecting the level of the RNA, and that's what's shown here. So you can see on GAPDH here, the RNA-Seq is unchanged with the taurin-1 treatment, whereas the RiboE-CLIP, you can see that diminution in ribosome loading on these transcripts, and that's shown transcriptome-wide here. And, we've compared RiboE-CLIP to, this is traditional polysome profiling, which again requires that sucrose gradient and fractionation, a really challenging technical endeavor. Versus RiboE-CLIP, which we've clipped multiple proteins from the ribosome and get equivalent data. And this is just showing the reduction in the ribosome load on the top motif containing genes following a taurin-1 treatment. So you can see that the RiboE-CLIP, with a relatively simple and straightforward protocol, can recapitulate the results that are seen with one of these traditional polysome profiling experiments. Okay, and I'm going to just finally, if I have time, I'm going to take you through CHAPE, which is a methodology that is used to probe RNA structure. So CHAPE, this is a technology that's been around for a while, and so there are multiple papers in the literature describing CHAPE. What we are doing at ECLIPS is really trying to make this technology accessible, sort of developing sort of services and kits around CHAPE that we plan to have on the market later this year. But again, this is a technology that's in the public domain, and I want to take you through what this does and sort of the implications more broadly. So CHAPE is selective 2' hydroxyl acylation analyzed by primer extension. It's actually, so that's the derivation of that acronym. And since it actually looks at the shape of the RNA, it's actually one of those acronyms that really works. So CHAPE at its core is really derived from treatment of using, I'm just going to call it NAI here, which is a chemical that can form adducts with the 2' hydroxyl of RNA molecules when that particular nucleotide is in a flexible position or accessible. So basically, you get that adduct formation when that nucleotide is unpaired. So I think you can sort of get the picture here that the adduct is really used to probe whether or not these nucleotides are in a constrained base pair position or in a flexible, more open position. And the interesting part about CHAPE technology is that you can apply it to cells. So basically look at RNA structure in vivo, or you can apply it to purified RNA in vitro. And I think the power of CHAPE can come when you do both in parallel. And that's really the direction that we're headed in terms of offering around CHAPE technology. So in the in vivo, the cells are treated with NAI and compared to a very important control. So this is just a carrier DMSO control. And these two are compared. Alternatively, CHAPE can be used on RNA that is either transcribed in vitro or isolated from cells, purified, and then refolded in vitro, and then probed with the NAI agent in vitro. So what this adduct does, I mean, there's a couple of different ways of looking at adducts in the context of NGS data. You can either look at the RT stops, or I think what's really a preferable method in the field is to change the conditions in which the RT is done, adding manganese to that reaction, which essentially causes the RT to incorporate a mismatch at a position where there's an adduct at an increased rate. That's quite a mouthful, but hopefully that's understandable. So what you see here is that this is the untreated control, and these little speckles here are the mutations. So manganese is causing a certain level of increase in that mutation rate by itself. But when you have the treated RNA that's been treated with the agent, you can see hotspots for these mutations. And ultimately, how this data is processed bioinformatically is that, mutation rates are calculated for the untreated, mutation rates are calculated for the treated, and then the comparison of the two is used to calculate a reactivity score. And reactivity scores can subsequently be fed into folding algorithms. There are many tools available online to do that. And these reactivity scores have been shown multiple times in the literature to really facilitate a better RNA fold than if you just did it simply with just the primary sequence. So that's what CHAPE is kind of in a nutshell. And, as I mentioned, this CHAPE data, really kind of the objective there is to really get a better sense, a more accurate sense of what areas of the RNA are, stably base paired versus relatively accessible. But there's additional variations on the theme, RNA genomics theme here, whereby the SHAPE technology can be combined with eCLIP for F-shape eCLIP, which is footprinting shape eCLIP. So this is essentially CLIP for an RBP combined with SHAPE, and this gives really nuanced data around the reactivity scores at that RBP-RNA interface. And in fact, in a recent publication by Corley et al. From Jean Yo's lab has shown that, this can actually illuminate the interactions down to the level of the hydrogen bonds at that interface. So there's a lot of powerful information that can be derived from a NGS experiment with the SHAPE structure probing. So finally, I want to just talk a little bit about the power of these technologies and, what it means when, you have all of these access to all of these technologies simultaneously. And as I mentioned, particularly in the context of the microRNA technologies I was talking about, with respect to getting deep data around particular target transcripts, gene enrichment, what we call gene enrichment, but actually we're enriching for an RNA of interest, can be employed to get deep data. But when you look at all of these different sort of orthogonal methodologies that can be employed, you really can start to get a multidimensional view of RNA. So again, that simple linear molecule or simple RNA-Seq data can really be transformed into a much more multidimensional view of RNA. And finally, I just wanted to give you just a little screenshot here of an IGV session and the power of all of these technologies together. Let me see. I don't know if I have time to pull up the IGV session, a live one, which I can do afterwards if anyone wants to stick around for that. But basically, just looking at this particular transcript, this is a relatively simple gene, BTG2 with 2 exons, and here's the 3' UTR. The End-Seq data, you get a 5' end call there. It's actually calling 2 different transcription start sites and a very precise single nucleotide 3' end call there that agree with the annotated transcript. Here's a heat map of the SHAPE reactivity, so you can see where there's hot spots for accessibility of that RNA. Here's your M6A and the individual single nucleotide M6A modification sites, which you can see that are really concentrated near that junction of the final exon with the 3' UTR. And that's a known hotspot for M6A modifications in the literature. And down here, ribo-eCLIP data showing ribosome occupancy and miR-eCLIP data. This is, actually you can scroll through each one of these. There's a lot of data in these sessions. This, has also all of the little individual microRNAs and their seed matching sites in this panel. And finally, in this particular session, we have all of the eCLIP ENCODE data, so you can look and see where, if any of those 150 RBPs that have been eCLIPed, if they are mapping to your favorite gene. So if anybody is interested to stick around after, I can pull up a live session and just kind of scroll around and kind of show you kind of the depth of the information that you can get from this technology. And so, yeah, I just want to end there and thank you for the invitation to speak in front of this group, and thank you for your attention. And I just wanted to mention that if you're interested in any of these, learning more about these technologies, please visit our website at eclipsebio.com. And also, if you found any of this intriguing, and I'm telling you, it's really exciting science, we're hiring at Eclipse in our R&D group at all levels, undergrad, master's, PhD, and postdocs. So do visit our careers page as well. Thank you very much.
Great, thank you, Karen. It's very informative. We do have a Curtis. You can go ahead and unmute yourself for a question.
Cool. Thank you. Such an interesting talk. I had a question about the M6A-seq because you said single nucleotide resolution. Is that because of the DRAC motif, that's metal 3's motif, which does do a primary amount of the methylation in the cell? I was just wondering if that motif was the reason for the single nucleotide resolution, and does that affect, because there's metal 16, metal 5, though they have only a handful of locations they introduce marks at, does that affect their resolution?
No, I think that the resolution is really kind of based on the biochemistry, I think. And
Okay.
So we're definitely seeing, M6A outside of the context of the DRAC motif, but it's just that we see it enriched there as we expect, right? So that Homer analysis on the data will, pull up the DRAC motif as the top motif, with a minuscule p-value. The single nucleotide resolution is really based on those cliffs that are formed in the reads that occur from that cross-linking of the antibody to the RNA. And, there was a lot of optimization around the biochemistry of that assay. Not all, I'll just say that, not all M6A antibodies are going to give you that really nice cliff. So, that's part and parcel of sort of the optimization that we've done on our end. And also there's, a bioinformatics pipeline that's really been optimized to process the data and get that single nucleotide call.
Nuria, would you like to ask your question?
Yeah, thank you. Yeah, that was a really nice talk. I'd like to know if the 5' seq or the 3' seq, for example, let's say like a 5' seq, are those quantifiable? Let's assume that I find out that there's a gene with like 2 TSSs, and under different conditions, like one is more active than the other. Is this really quantifiable?
Yeah. So it's, it's similar to an RNA-Seq. So, the number of reads mapping to those particular start sites. So basically the height of the peak at those different start sites is quantitative.
Okay, thank you.
I was just curious when some of these new technologies you mentioned, like the RiboECLIP, when would that expect that to be available?
So we're hoping to launch, over the next couple of months, actually. So, both services and also an easy to use kit.
Great. Are there any other questions? You can unmute yourselves if you'd like to ask.
Yeah, hi, this is Matteo Vatta. Very interesting talk, and I have a question about the technologies that you have showed in your in details in your presentation have been used to identify constitutional variants that can affect, for instance, long-range non-coding RNAs or even microRNAs that will show a different effect of binding to messenger target that could be potentially linked to human conditions. So for instance, just to give you an example, there are microRNAs that have been identified in cardiovascular diseases, and they have multiple targets. But there are very few analyses of variation, human variation in those microRNA sequences, although very small, of course, but they may have variations in their alter pattern of target, binding and modulation of the transcription.
Oh, that sounds really interesting. And I think that miR-eCLIP technology could be really helpful there because you get the actual sequence of the microRNA linked to the target, and you could deconvolute if there were mutations in the microRNA. But to my knowledge, I haven't I'm not aware of any studies like that. It's a relatively new technology. In fact, we're just prepping the publication on it now. So, stay tuned for a publication sort of detailing those the whole microRNA and the enrichment technologies I discussed. So I think, for a lot of these technologies, the biological implications haven't really been, fully digested. And I think it's really when the technologies become more available and accessible that you start to get that explosion of biological relevance.
Have you tried, for instance, mutagenizing in vitro some of the microRNA just to see, you have, let's say, like a internal control of the technology itself is working and is quantitatively relevant.
Have we mutated RNAs?
So for instance, for the MCA, mutagenizing the adenine in order to change the nucleotide could alter, of course, the methylation of the adenine, and therefore could alter the binding and the assay itself.
Right. So I don't think that we have done that particular experiment, but I didn't go through all of the data sort of validating the assay, and there's quite a bit. And there's different M6A technologies out there as well. So, I think there's quite a foundation around M6A and the validation of, antibody-based methodologies.
Thank you. Well, the background reason is that we have seen recently how modifiable organism with RNA, like the COVID virus, is modifying its sequence so rapidly and so efficiently, and therefore having a technology that is flexible enough to identify also variability due to naturally occurring variants on RNA target is very important.
Agreed.
I just had one question in the chat. Can the RiboE-CLIP be used with the RiboTag?
RiboTag? Yeah, I'm unfortunately not familiar. Oh, do you mean, I'm not sure exactly what you mean by a tag.
So hi, I'm Brian Percella. I asked the question. So RiboTag is basically a genetically encoded HA tag.
Oh, okay.
So it's a way of purifying cell type specific ribosomes and then sequencing the RNAs bound to them. But it requires an HA antibody for the IP. So in other words, could you substitute an HA antibody for RiboE-CLIP and then just do the eCLIP steps after that?
I would imagine that you could. Yeah, we've definitely done that in the context of eCLIP. So I would think that would work.